Hello everyone, myself Birupakshya Choudhury, research scholar from the Department of Life Science and Bioinformatics, Assam University, Silchar. At the outset, I would like to thank my supervisor, Dr. Choudhury sir, Assistant Professor in the Department of Life Science and Bioinformatics, Assam University, for providing me this wonderful opportunity to be a part of the great initiative taken by the Zoological Society of Assam to help the undergraduate students of zoology and life sciences in the harsh time of pandemic. The topic on which I am going to deliver is the history of cell discovery. Although presently we know a lot about the cells, their function and behavior, but it is really fascinating to look back towards the historical perspectives of cells and how they were discovered. So let us start. In this video, we are going to cover the following topics. What is a cell? The invention of microscopes, discovery of cells, under which we will discuss the cell theory, protoplasmic theory, organismal theory, and the molecular theory of living cell. After that, we'll briefly discuss the origin of cells, and then we'll see some of the important discoveries of cell biology which received Nobel Prizes, and before concluding, we will have an idea about the branches of cytology. So let us come to the topic. What is a cell? Now this question we have been answering from our school days, that cells are the basic building and control units of life. But what makes them so special and important? This is because cells are those entities which not only build one organism but also control and regulate all the vital life-sustaining functions in an individual. So on the basis of the presence or absence of nucleus and other membrane-bound organelles, cells are either eukaryotic or prokaryotic respectively. Interestingly, there lies many similarities in various processes in both these types of cells, and hence it is considered that both of these types have originated from a common ancestor. Cytology is that branch of science that studies cells and its structure, physiology, division, death, and various interactions between cell and its environment. Today, if you ask the definition of cell to a school student, she or he may easily answer because it is an established fact now. But just imagine a condition hundreds of years back when people had no access to microscopes or any other advanced technologies. For them, the terms like cell or cell cycle or nucleus or even genes were all incoherent, because no one had actually ever thought that something which is so minute and cannot be seen with unaided eyes may be the unit of life on this planet. But what actually changed the whole scenario was the invention of microscopes. So the turning point in cell discovery and many more parts of biosciences was actually the invention and gradual development of microscopes. The current knowledge of cell biology is the outcome of the works by a large number of scientists across centuries. Since cell is too small to be observed with naked eyes, it was not possible to see or study or even know about its existence without the help of magnifying instruments. The invention of microscope was therefore a boon to the discovery and for the study of cells and the miniature life forms. Invention of microscopes paved the way for various branches of studies such as cytology, microbiology, etc. Let us swiftly move across the various events which had led to the invention and gradual development of microscopes, for which today we have some of the best imaging techniques in science. The first kind of such invention dates back to 1284, when Salvano Armati invented wearable eyeglasses. In 1485, Vinci recommended the use of lenses in viewing small objects. In 1558, Conrad Gessner published the results of his observation of protists called foraminifera. It was obvious from his sketches that he had used some form of magnifying lenses. 1590s, a remarkable period when Hans and Zacharias Janssen, the father-son duo, designed the first compound microscope having 3 to 10 times magnification. During the same time, Hans Lippershey is also attributed to the development of microscope. In 1609, Galileo improved the device further and named it Occhiolino, which means little eyes. Galileo's invention was termed as microscope for the first time. During the 1660s, Robert Hooke improved the magnifying ability of microscopes and became the first person to observe cells. Here we can see Robert Hooke's self-designed microscope, which is described in his book Micrographia. During the 1670s, Dutch scientist Antony Leeuwenhoek developed his own microscope and became the first person to observe living cells through his lenses. The illustration of Leeuwenhoek's microscope is given here. In 1826, Joseph Lister modified a chromatic lens, thereby eradicating the chromatic effect. Now, chromatic effect is a phenomenon when a lens fails to focus all colors of an object to the same point, and this mostly happens due to the problem of refractive index of the lens. In 1878, physicist Ernst Abbe designed a modern achromatic compound microscope having illuminating system and condenser. During the 1930s, another remarkable period when Knoll and Ruska developed the electron microscope, which proved to be a milestone in the field of microscopy. In 1986, Ruska was awarded Nobel Prize for this achievement. In 1932, Zernig developed the phase contrast microscope. Basically, phase contrast microscope is used to study the colorless or transparent specimens. In 1957, Minsky developed the confocal imaging technique. In the year 1978, Thomas and Christoph Kramer developed the confocal laser scanning microscope. In the confocal imaging technique, laser light is focused onto a definite spot at a specific depth within the sample, and it does not illuminate the whole sample at once, which the conventional microscopes do. During the 1990s, we finally came across the fluorescence microscopy. So here you can see the picture of a typical electron microscope. So here we discuss the discovery of cells and the role of Robert Hooke. It was Robert Hooke who actually discovered the cells. Robert Hooke was an English physicist and a fellow of the Royal Society of London. During 1665, with the help of Christopher Koch, an instrument maker, Hooke designed his own microscope, which later came to be known as Hooke's microscope. With this instrument, Hooke observed some honeycomb-like structures in a section of wooden cork, which he termed as cells. Because the spores or honeycomb-like structures reminded him of small rooms in which monks reside in monasteries, and in Latin, cella means small rooms, that is why he termed the structures as cells. Actually, the structure that Hooke had observed were the empty cell walls of the dead plant tissue which made up the cork. Robert Hooke described his observations of tiny and unseen world in his famous book Micrographia. So here are some of the illustrations from Micrographia. On the left, these are the illustrations of cells which Hooke had observed from the section of wooden cork. In the middle, this is the sketch of Robert Hooke's microscope, and on the rightmost side, it is the illustration of a flea which was observed under Hooke's microscope. Here, we discuss the role of Antony Leeuwenhoek in the discovery of cells. Antony Leeuwenhoek was a Dutch cloth merchant who came across magnifying glasses used in cloth shops to observe the thread densities. Leeuwenhoek somehow became interested in studying microscopic life forms, for which he designed his own simple microscope having 200 to 300 times magnification. In 1676, Leeuwenhoek became the first person to observe microscopic creatures in drops of pond water, which he termed as animalcules. Next year, that is in 1677, Leeuwenhoek described spermatozoa for the first time and postulated that sperm and ovum combine to initiate pregnancy. Leeuwenhoek also studied plant cells, human cells such as muscle, skin, and even the scrapings from teeth. First representation of bacteria is found in a drawing by Leeuwenhoek, which was published in 1683. Since Leeuwenhoek was the first person to observe the presence of miniature life forms, he is sometimes considered as the father of microbiology. So here we come across the roles of Schleiden and Schwann in the discovery of cells. In 1838, Matthias Schleiden, who was a German botanist, observed that plants were made up of one or more cells. In the following year, Theodor Schwann, a German physiologist, also found that animals were made up of cells and that both plants and animals had similar structural units. They collaborated their ideas and proposed that cell is the structural and functional unit of all living organisms. Cell is a distinct entity, that is, cells can live independently as unicellular organisms and also are the building units of all organisms, and that cells originate like that of crystals. These propositions are known as cell theory. However, Schleiden and Schwann's idea about origin of cells are less insightful, as because somewhere this idea was influenced by the theory of spontaneous generation and there was no basic understanding regarding the origin of cells. Since Schleiden and Schwann were unable to address the exact mechanism of origin of cells, the third tenet of cell theory was not widely accepted. In 1846, Carl Nageli showed that plant cells can arise from the division of pre-existing cells. In 1855, Rudolf Virchow, a popular pathologist, confirmed this view regarding cell division, and also from the findings of Robert Trimac, famously propounded omnis cellula e cellula, which means cells arise from the division of pre-existing cells. This statement is widely accepted as the third tenet of cell theory. Later in 1865, Louis Pasteur provided experimental evidence to Virchow's extension of the cell theory. Depending on the developments of science and technology, the original cell theory has been developed into modern cell theory. So let us see what are these. Cell is the structural unit of life. All living organisms are made up of one or more cells and cell products. Cells originate by the division and duplication of existing cells. All metabolic reactions in unicellular and multicellular organisms take place in the cells. The significance of such theories lies in the greater scientific developments which had helped the scientists to uncover the details of cells and their functions. The classical theory, although describes the origin and structural significance of life, the modern cell theory includes the physiological and genetic basis of cells. The protoplasm theory. So basically there were a sequence of events which had highlighted protoplasm to be an important entity in cells and life altogether. In 1835, Dujardin termed the jelly-like material within protozoans as sarcode. In 1839, Purkinje coined the term protoplasm to describe the contents of cells. In 1846, Moll applied the term protoplasm to the contents of plant embryonic cells. In 1850, Kohn demonstrated that protoplasm of plant and animal cells were similar. In 1861, Skurge suggested that protoplasm is basis of vital activity in living organisms. In 1868, Huxley suggested that protoplasm is the physical basis of life. And in 1892, Hartwig proposed the protoplasmic theory, considering its importance in life. So basically, protoplasm theory postulates that the physical basis of life, including the foundation of heredity, must be sought in the properties of this universal biological substance. The last quarter of 19th century is usually considered as classical period of cell biology. The organismal theory. So basically this theory was developed as a counter argument to the cell theory and protoplasm theory. The theory was based on some observations. Reichardt, a morphologist, observed that organisms have their own structural plans. Some embryologists observed that early chordate embryos have free nucleate stages. It was also found that some animal tissues have no cellular boundaries. Edward Strasburger observed that cells are connected by cytoplasmic bridges and was thought to be a continuum of protoplasm instead of separate cells. Sir Charles Sherrington and Ivan Pavlov found that cells communicate with each other and coordinated in their actions. Wilhelm Roux, an embryologist, observed that destruction of a cell from a two-celled embryo led to the formation of a partial embryo. These findings suggested that significant parts actually constitute an organism rather than cells. So these observations laid the foundation of organismal theory, which states that the body of a typical multicellular organism is a continuous mass of protoplasm which remains incompletely divided into cells. So this theory actually popularizes that organisms have their own complex organization, structural features, and functional abilities apart from those of the constituent cells. But currently it is assumed that most of those ideas grew either due to erroneous observations or due to the lack of clear understanding of molecular mechanisms. For better understanding of the cell and organismal doctrines, let us go across a simple diagram which will make it clear. So, as per the cell doctrine, cells are considered as recognizable system which supports itself and contributes to an intercellular pattern. But in organismal doctrine, cells are considered as units which contribute different features to an overall organismal pattern. As the scientific world experienced a boost in technology and greater insights into molecular mechanisms of matters, cell biology also reached its heights. Scientists have henceforth observed the origin and development of life with a new and different perspective. In 2012, Choudhury, a theoretical cell biologist, proposed the molecular theory of living cell. According to him, this theory is an effort to contribute towards the rise of the new science of human nature. This theory, which we will discuss shortly, is based upon almost 30 concepts, including the principles and laws of thermodynamics, statistical and quantum mechanics, chemical kinetics, informatics, computer science, linguistics, semiotics, and philosophy. By the way, semiotics means the study of signs and symbols and their uses. So as per the theory, the cell is an organized system of molecular machines, that is, the biopolymers, which are DNA, RNA, proteins, and which carry out microscopic work processes, including enzyme functions, cellular transport, molecular motor movements, gene expression, repair, and self-replication. But the main part of this theory depends on conformants, which are packets of mechanical energy stored in certain sequence-specific sites within the biopolymers and are derived from the chemical reactions based on Frank-Condon mechanism. Now let me say here, Frank-Condon mechanisms are related to spectroscopy and quantum chemistry, which explain the simultaneous changes in electronic and vibrational energy. Therefore, the living cell is a supramolecular machine driven by chemical reactions mediated by conformants. So schematically, this theory can be represented in the following way. So as I said earlier, this theory is based on the existence of conformants, which are packets of conformational energy generated from substrate binding and chemical reactions. To understand the conformants, we may take the example of a piano, where the conformants can be compared to vibrational energies generated by the vibrating strings. Here we will swiftly go across various important events in cytology. So in 1802, Franz Boyer pointed out the presence of nucleus in cells. In 1831, Robert Brown introduced the term nucleus for some opaque spots on the cells. In 1835, Dujardin discovered the jelly-like substances in cells which he termed as sarcode, but these were actually protoplasm, which were coined by Purkinje in the year 1839. In 1848, Hofmeister discovered chromosomes. In 1857, Albert Kolliker discovered cytoplasmic organelles, which were later found to be as mitochondria. In 1863, Kolliker coined the term cytoplasm. 1865, a remarkable year for science when Gregor Mendel published the results of his experiment with pea plants, which became the basis of genetics. In 1869, Friedrich Miescher discovered and coined the term nuclein, which is isolated from the nuclei of pus cells from patients. Later on, Miescher's nuclein was found to be the nucleic acid. From 1879 to 1882, Walter Fleming extensively studied the cells and introduced the term chromatin and mitosis. In 1888, Waldeyer Hertz introduced the term chromosome. In 1888 itself, Theodore Boveri proposed the role of chromosomes in heredity. In 1891, Waldeyer introduced the term neurons to indicate nerve cells. In 1898, Carl Benda introduced the term mitochondria. In 1898, Camillo Golgi discovered the Golgi apparatus. In 1901, Edward Strasburger introduced the term plasmodesmata. In 1902, Boveri and Sutton proposed that chromosomes bear hereditary factors. In 1905, Farmer and Murray coined the term meiosis. In 1909, Wilhelm Johansen introduced the term gene for the hereditary units. In 1910, Morgan established the chromosomal theory of heredity. In 1913, Sturtevant created the first gene map. In 1935, Danieli and Davson proposed the sandwich model of plasma membrane. In 1943, Claude isolated cellular components such as ribosomes, mitochondria, nucleus in pure form by differential centrifugation. In 1951, HeLa cell lines, which are cancerous cell lines, were established from the cervical tumor taken from Henrietta Lacks. These cell lines are immortal and are still used in research. 1953, a very important year for bioscience, when Crick and Watson deduced the structure of DNA. Findings of Rosalind Franklin and Maurice Wilkins were also very important for this development. In 1955, lysosomes were described by Dewey. In 1959, Robertson introduced the unit membrane concept. In 1960, Brenner, Crick, Jacob, and Monod discovered the messenger RNA. In 1972, Singer and Nicholson proposed the fluid mosaic model of plasma membrane. In 1981, Lynn Margulis proposed the endosymbiotic theory to describe the phenomenon of symbiosis in cell evolution. In 1998, James Thomson isolated human embryonic stem cells and developed cell lines. In the year 2000, the human genome was sequenced, and in 2001, its reports were published. The Human Genome Project was an international project undertaken by the International Human Genome Sequencing Consortium. So let us briefly discuss the origin of cells. It is considered that life originated 3.8 billion years ago. It was first suggested in 1920s that the simple organic molecules could have polymerized into macromolecules under the primitive conditions of Earth, during which there were little or no oxygen in the atmosphere. The first cell is presumed to have arisen by enclosing a self-replicating nucleic acid molecule within a phospholipid membrane and had maintained the ability to reproduce and proceed towards evolution. Eukaryotes developed around 2.7 billion years ago, that is almost after 1 to 1.5 billions after the advent of prokaryotic cells. There are certain evidences which suggest that all present-day cells have arisen from a common prokaryotic ancestor. The endosymbiotic theory of Lynn Margulis suggests that eukaryotic cells have developed from a symbiotic association with other prokaryotic cells. And this has been also supported by the molecular studies of mitochondria and chloroplasts, which show evidence that these were once upon a time free living organisms like that of a bacteria. These are some of the Nobel Prize winning discoveries in cell sciences, although many more are there. Interested students may check out the introductory pages from Cell and Molecular Biology by Gerald Carr. Some of the discoveries stated here have been also mentioned earlier. So in 1906, Golgi and Kagel received Nobel Prize for developing methods that color and highlight the nervous system. In 1930, Karl Landsteiner received Nobel Prize for discovering human blood groups. In 1974, Claude, Diouf and Palade received Nobel Prize for discovering the structural and functional organization of the cell. In 1989, Bishop and Vermus received Nobel Prize for discovering the cellular origin of retroviral oncogenes. In 1993, Thomas Morgan received Nobel Prize for discovering the role of chromosomes in heredity. In 1994, Gilman and Rodbell received Nobel Prize for discovering G proteins and their role in cellular signal transduction. In 1996, Doherty and Zinkernagle jointly received the Nobel Prize for discovering the specificity of cell-mediated immunity. In 1997, Prusiner received Nobel Prize for discovering prions, an infectious agent. In 2001, Hartwell, Hunt, and Nurse jointly received Nobel Prize for discovering the key regulators of cell cycle. In 2002, Brenner, Horvitz, and Sulston received Nobel Prize for discovering the genetic regulation of organ development and programmed cell death. In 2008, Hausen received Nobel Prize for discovering human papillomavirus as a cause of cervical cancer. Sinosi and Montaigne also shared this prize for discovering human immunodeficiency virus or HIV. In 2012, Sir Gurdon and Yamanaka received Nobel Prize for the discovery that mature adult cells can be reprogrammed to become immature pluripotent stem cells, which can give rise to many types of cells. In 2013, Rothman, Shackman, and Shadoff received Nobel Prize for discovering the cellular transport systems. In 2014, O'Keefe, May-Britt Moser, and Edward Moser received Nobel Prize for discovering the cells that constitute a positioning system in the brain. In 2016, Osumi received Nobel Prize for discovering the mechanisms for autophagy. In 2019, William Kaelin, Sir Peter Ratcliffe, and Greg Semenza jointly received Nobel Prize for their discoveries regarding how cells sense and adapt to oxygen availability. Come across the branches of cytology. So let us see what are the various branches of cell studies. Cytochemistry, which deals with the biochemistry of cells. Cytopathology, the study of diseases at a cellular level. Cytotaxonomy, branch of taxonomy that uses cytological structures, especially the chromosomes, to classify organisms. Cytoecology, branch of cytology that studies adaptive reactions of organisms at the cellular level. Molecular cell biology studies the interaction of molecules that results in various cellular properties. Cell physiology is the study of cellular function and its relationship with the surroundings. Cytogenetics is the study of chromosomal structure, location, and function in the cells. So in conclusion, I would like to point out that the discovery of cells not only unveiled a wide array of microscopic world in front of us, but also enabled the human civilization to understand the origin and evolution of life on this planet. Since cells are the basic unit of all life forms, it is very important for us to understand the cells before we go in depth to other biological processes. Lastly, we are thankful to the researchers working very hard to solve many biological mysteries and bring out the answers of such puzzles to the eyes of humanity. And before winding up, there is a beautiful message for the young minds who are frequently depressed because of various factors. So basically, there is no need to be unhappy or distressed because all the cells of our body are continuously working to keep us safe and healthy. So cheer up. So these are some of the references that have been used to make this video. Students may go through the books of Cooper, Karp or Bruce Alberts because these are very good books for cell biology and they will get whatever they need in those books. And here are some URLs which have been also used to gather information regarding this topic. So thank you very much, and I hope I was able to cover up most of the things which is associated with this topic. If anyone is having any sort of doubts or confusion, please don't hesitate and you may contact me in the email ID which I have provided. Thank you and happy studies.